None of the content on this or any episode of the Kratom Science Podcast and Kratom Science Journal Club, nor on any of the pages of KratomScience.com, should be considered medical claims or medical advice. This is the Kratom Science Journal Club with Dr. Jonathan Cachet, neuroscientist and expert in psychopharmacology. In each episode, we discuss an article in a peer-reviewed journal. I'm your host, Brian Gallagher, blog and social media writer for KratomScience.com, your source for all things Kratom. Episode 26, we're looking at another study from University of Science, Malaysia.
I work down at the University of Science there.
Cognitive impairment in mice were found with chronic high-dose mitragynine, similar to morphine and THC, unless the mice were also given a CB1 receptor antagonist. So this is called mitragynine, parentheses, kratom-induced cognitive impairments in mice resemble THC and morphine effects, delta-9 THC and morphine effects, reversal by cannabinoid CB1 receptor antagonism. And this is from the journal, I guess it's Frontiers in Pharmacology. And this is a team at University of Science in Malaysia.
I work down at the University of Science there.
Our good friends at University of Science. So it's a mouse study, as it says in the title. And it pretty much says everything in the title. The mice were given low and high dose mitragynine, THC, and morphine, and then four other sets were given all these drugs with a CB1 antagonist, which is called NIDA 41020, and is that because it was developed by NIDA?
Yeah.
Okay.
Yeah, there's a lot of especially cannabinoid receptor agonists and antagonists that are just like I think Win 1500 is another example of one. But yeah, they're just analogs or closely resembling them and activity at those receptors.
In this study, the involvement of cannabinoid type 1, CB1 receptors, and cognitive deficits after chronic mitragynine exposures was investigated for 28 days. And I think it was 100 adult male Swiss albino mice using the IntelliCage system, which I looked that up, and it's basically a high-tech, sciencey cage. On their site, it says it fosters natural social behavior in a standardized home cage context, minimizes the need for handling and human intervention, increasing task validity, data reproducibility, ensuring high level of animal welfare, well, not in this case because they euthanize them after the study.
Ultimately, yeah.
Study their brains, and they all go to mouse heaven. And they have those RFID tags, radio frequency identification tags, so it goes right into the science machine, and they could study that.
Right into the calculator attached, yeah. So these cages are really, I want to say modern marbles, but they are really something that reduces the amount of handling and allows for automated data collection over a number of different endpoints that can be interpreted in different ways, which I think that this paper really lays out a good use case, because we're talking about 100 mice, 5 different treatment groups. We're doing a 30-day month protocol from the implantation of the RID tag, so you can ID individual mice, all the way through to sacrifice. So without the IntelliCage and that technology, this would require someone marking it with a notepad and a piece of paper, and maybe multiple people every day for all of those 30 days. And the data certainly wouldn't have been as dense as it is, which I think is a good study design. And they went from the psychopharmacology, the behavior, all the way through to the immunohistochemistry, and that's all laid out in this one paper. So overall, I think it's pretty impressive.
Yeah, it was pretty comprehensive, and And just the one thing that I kept seeing is even in the title it says Mitragynine Kratom, but they're using Mitragynine alone and not Kratom. So they, why do you think they don't use, like, in that one study, they use, like, lyophilized Kratom tea that contained, like, the full spectrum alkaloids or, like, a full spectrum alkaloid extract? Why do you think they just use Mitragynine? I guess because they're studying pain or whatever.
Well, I think in this case, you're limiting the amount of confounding variables that you want to introduce to this. So we already have, like, multiple pharmacological treatments, the use of an antagonist, multiple treatment groups. And so if you add on top of that, like, full spectrum kratom extract, which is, made from leaf into like a tea or whatnot, it's not very standardized. And so, yeah, the confusion there of, like, saying, well, mitragynine is kratom is sort of misleading, but I think that their stated goal was to elucidate mechanisms behind like potential exploratory behavior or cognitive impairment or learning and memory. And when you have a study of this, from behavior through immunochemistry and genetic expression, you just want to limit your potential vectors of variability or variance.
And they found with the chronic high-dose mitragynine exposure, but not the low-dose exposure. And this is all, like, injected into the abdomen. It potentiated preference for sucrose reward, increased resistance to punishment, and impaired place learning and its reversal. And it says the same response was with THC and morphine. And this is all due to its action on the CB1 receptor. We know what THC and morphine does. Kratom, has never done that same kind of thing. Like, does it just mean really that they're high? Is that basically what's happening?
That's a good question. Yeah, and you really to really digest everything that's going on in this paper, you have to sort of go through the results section and the figures one by one. Mm-hmm. In that, at first they're establishing a baseline. So inside of these IntelliCages, there are corners that have access to water, and they placed 20% sucrose solution in one of the other corners. And so for the first couple of days, you just put the mice in there and establish their baseline activity. So what do they just do in this novel environment? And what first jumped out to me, or at least the difference between the control groups and the treatment groups, was we had high-dose mitragynine and morphine leading to elevated hyperlocomotion. So they were moving more. They were exploring the cage more frequently. And so you don't necessarily call that a cognitive deficit. It's sort of like a weighted term to say that something's a deficit. So they have more activity, whereas the THC ones had less activity. So that's why they were constantly comparing baseline to then later in the study. But where the interpretation and the study on where they were looking at cognitive deficits was sort of more probably more specifically designated as how quickly they were able to learn where something was, and then when they moved that something, how quickly they were able to realize it was moved and the new location. Does that make sense? Is that how you read it as well?
Yeah. Is that the place preference one? Okay. Yeah. I just kind of read it as they just had a harder time learning in general, I guess. With the morphine. So CB1's a cannabinoid receptor, and THC is an agonist. And so I guess apparently morphine and mitragynine also act on cannabinoid receptors, which I don't think is something we talked about before.
And I don't necessarily think that there's a direct interaction there either. There's a lot of overlap with reward centers and motivation and anticipation and planning of behaviors. And I would guess sort of cognitive switching between different tasks at hand. But what they're what they're showing with this set of studies overall, behaviorally at least, is that if you block the activity of CB1 receptor, then the presumable reduction in learning time or ability to learn where the sucrose was, the place preference, that was no longer found. So like you block CB1 and the longer time or higher percentage of incorrect guesses that the mice made that were in the treatment group was no longer evident. So that's, a mechanistic explanation for how, and I don't want to say cognitive deficits. When I say cognitive deficits, and in this paper, what they mean is it took them longer to learn, and it took them longer to unlearn. And so, by showing that affects across different drug groups was modified or eliminated with the CB1 agonist, then they're demonstrating a role for CB1 receptors and CB1 receptor activity in the acquisition and subsequent relearning of these tasks.
Okay, and yeah, it also said significant upregulation of CB1 receptor expression was found after chronic high-dose mitragynine and morphine, whereas a downregulation was observed after chronic THC. So what does that mean?
Well, so I can speculate. I don't want to sort of dig into the results section a little bit more, but essentially one of the mechanisms proposed behind tolerance and dependence is that if you expose the neurons to exogenous high concentrations of morphine, the morphine receptors, there's several different subtypes there. We're talking about CB1 here, but I'm just using the analogy to get us there, is that you develop a tolerance and dependence because the constant overactivation of those opiate receptors leads to them being pulled into the cell. And remember, that was what do we always talk about, where it's a mediated response of that cellular induction, and it's like a kinase, or not an interleukin, but kratom and mitragynine doesn't activate those in the opiate receptor system, so we don't see that downregulation. I forget what the exact, we talk about it in a lot of the journal clubs though, about it doesn't activate this pathway, so it doesn't lead to the induction of these cells. So specifically here, they see an upregulation in the hippocampal, so that's sort of learning and memory, and VTA is sort of vigilance and alertness after a chronic high dose of mitragynine and morphine. So there are more CB1 receptors expressed after chronic mitragynine use and morphine use, whereas a downregulation or less receptors was observed after chronic THC. And so if you relate that back to what I was saying, chronic THC, delta 9 THC does act directly at the CB1 receptors. And if you're exposing those receptors to the THC, repeatedly over a long amount of time, it leads to an induction of them into the cell. So a downregulation of those receptors. That's analogous to opiate receptors in morphine. Now, why would there be an upregulation of the CB1 receptors as it relates to mitragynine or morphine? There's got to be a set of skipping stones here that they probably get into, and I think they do get into in the results and discussion. But essentially, the activity of these drugs at the primary receptor led to either less activity at the cannabinoid 1 receptors, or less availability of agonists for those receptors, and so it led to the cells expressing more. It's helpful to consider all of these biological systems as machines that are just designed to maintain homeostasis and responding to a teeter-totter effect of is there more, is there less, how can we establish balance again? Mm-hmm. So what's eluding me in the part that I didn't actually have them connect, and this maybe is because it's buried within the discussion, is essentially if they're seeing that high chronic dose mitragynine and morphine and chronic THC cause these impairment of their ability to learn the place preference and then relearn the place preference, wouldn't we expect the upregulation or downregulation of the CB1 receptors to be the same? And because it's not the same, that points to a different set of mechanistic skipping stones as to why the effect is there. Like, we ultimately know that the CB1 receptor is responsible because we can extinguish the effects of the slower learning by blocking the activity of the CB1 receptor. But that doesn't necessarily explain to me the details of why you see it go up in the opiate or sort of narcotic category and down in the delta 9 THC category.
It was just another question related to mitragynine alone. Could other alkaloids maybe inhibit its activity on the CB1 receptor, other alkaloids in kratom?
There could be, yeah. But that would be, I mean, that's why they probably didn't go with the full spectrum extract, because then they wouldn't have been able to like, definitively answer that. Now they just know that mitragynine's there. And so I thought I remember reading deep into the discussion, and I'm trying to look for my highlights in my notes here. I don't know exactly where it was. Let's see. The mitragynine-induced receptor upregulations appeared to be attenuated by the antagonist, as seen in the immunohistochemical protein and mRNA studies. These findings suggest a potential mechanism for the beneficial effects of CB1 receptor agonism at the behavioral level. And present results may also indicate a plausible chronic mitragynine CB1 receptor interaction in inducing the transition from liking to wanting response. And so they're talking about behavioral sensitization and a theory underlying addiction behaviors there.
Mm-hmm.
Let me just finish this. Transition from liking to wanting response, which eventually culminates in the development of mitragynine/kratom addiction. So I don't know. I guess that speaks to what we're sort of talking about here, but I don't think it necessarily provides a definitive answer, and nor do I think necessarily that they were. I think that they were just trying to establish a role of the cannabinoid receptor in these behavioral impairments, not necessarily like they could do a more focused study now and perhaps get to that answer.
Okay, so they euthanized the mice. They looked at they took brain samples. Now, the first thing they said is the immunohistochemistry to investigate CB1 receptor expression. Now, what does that mean exactly?
So the immunohistochemistry, so that staining was performed to investigate CB1 receptor expression. Is that what you asked about?
Yeah.
They are euthanizing the mice, usually with pentobarbital and then like a cut to the neck. And then they very quickly dissect the brain out. And then they put the brain they're blocked in paraffin. Paraffin is like, consider it just to be like epoxy. It's not just epoxy. It's obviously like designed to lock in the brain structure and allow you to cut thin slices of the brain structure, but it also like doesn't react with the neurons or the other like the biological tissue in any way. But the immunohistochemistry would be you extract the brain, you put it in the paraffin, and you also expose it to antibodies. And so there are antibodies that will detect the CB1 receptor. When you expose those antibodies there, the more CB1 receptor there is, the more staining will occur because the stain is attached to an antibody that specifically binds to the CB1 receptor. So that's all they're doing there is seeing like if I expose it to this stain and the stain only appears in areas where CB1 receptor is, then more or less stain would indicate more or less of the CB1 receptor at that particular location.
And then Western blot, what is that?
A Western blot is along the same lines as immunohistochemistry, but it's not in situ. It's like not in brain tissue.
Okay.
So in these scenarios, it's gel electrophoresis, and you basically like extract the compounds of interest from the tissue sample. You put it at the top of what is like almost like a Jell-O pad. This is a gel electrophoresis. It's obviously, of course, not Jell-O, but it's very similar. And you put a positive current and a negative current at either end of this pad, and then you watch how the proteins migrate through that pad. And depending on like how polarized they are, the size of these proteins, and the amount of amperage or voltage that you're hooking up to these probes, will depend on how much it slides down. So like when you see the results that show like a line in, let's see, looking at, page 14. So like figure 8A, those lines are the blots in the gel electrophoresis, or the Western blot, right? This was Western?
Yeah.
It's been a minute since I have been in the lab doing gel electrophoresis. There's a difference between a Western blot and like an Eastern blot, and I think there's other variants in between. I don't. It's not clear to me exactly what that is. There is a difference there.
Okay, and is this is like looking at like genes and stuff, like DNA? Is that what that is?
Yeah. So you can look at DNA fragments.
Yeah.
You can also look at proteins. You can look at a number of different things in electrophoresis.
And that's basically what quantitative real-time PCR does too. It looks at the DNA.
Well, so quantitative real-time PCR is much different than gel electrophoresis in terms of how it's like the actual mechanism. So quantitative PCR is polymerase chain reaction, but essentially, if there's a DNA fragment or several DNA fragments related to, let's say, the CB1 receptor being expressed in the tissues at that time, you put in specific enzymes and primers into the quantitative PCR, and any of those fragments that are exposed essentially result in the replication of that DNA. So you amplify or like induce replication of specific fragments that you're interested to know how much is being expressed inside of the tissue. And the quantitative part is basically like you take it from a concentration where it's like, let's just say 1-fold, you go 1,000-fold, and based on like the actual numbers of going from 1 to 1,000 and the amount of time that it took, you can make an assessment of the availability or how much of that RNA was currently being expressed in that tissue sample at that time.
So they will go after sucrose instead of water more frequently when the mice take high dose mitragynine, morphine, or THC. Is it that the drugs, when they're administered, kind of train them to be more reward seeking?
Like more hedonistic about the whole situation. I think that's the justification.
Yeah.
I think the idea, is that they are taking drugs that sort of enhance pleasurable activities, and these drugs must have some pleasurable like basis to themselves because they're they're like seeking more of that pleasurable behavior, being more hedonistic. I also sort of share with you, I think if I'm sensing it right, just a little bit of skepticism where it's like, okay, well, you want to talk about, at the front and the bookends of the paper where like kratom is a widely abused plant. It was sort of like drug hysteria a little bit. And so then it makes me sort of say, okay, so they went to go get sucrose more than what they normally would have, but the other mice were also, hitting that sucrose. Is that really indicative of all three of these compounds just at face value are, horribly addictive or like alarmingly addictive? I don't know if that necessarily makes that case.
Yeah, I mean, for the THC, it could be because they have the munchies or something.
That's what I was thinking. Or they have dry mouth and they're just trying to get them to drink more.
And really, like the cognitive impairment, I've never experienced any type of cognitive impairment on kratom. They said something about mitragynine and other studies showed some kind of emotional impairment, which I think I've been like extra irritated because I was on kratom, but I mean, I would have already been irritated, but it just kind of upped it, and I've seen that in some people, like on Twitter and stuff. but it's never been like, it's never prevented me from learning anything or reading, or it just seems like, whereas like, marijuana, or maybe like, I haven't done enough opioids to have an example from personal use, but whereas like marijuana, I probably wouldn't smoke pot the first day I'm going into a new job and learning, being trained. I wouldn't. Right. I would want to be sober.
the data that supports them saying that there was like a cognitive impairment is just it's pretty well laid out in figure 4. Okay. So there's the place learning and then reversal learning. And you can see, and I think this is right, but I was trying to see. So like the THC is that purple upside down triangle at the very bottom. Mm-hmm. Morphine is the lime green with the right side up triangle, and then mitragynine high is the orange. And so the y-axis is percent of correct or percent of preference for correct corner, meaning the corner in which they're getting the sucrose reward. Mm-hmm. So these mice in their place learning phase were more frequently incorrect in going back to the water bottle that had the sucrose in it. Whereas like you could see on all the other lines that sort of fade up to the top, like once these mice realized where the sucrose was, they pretty much just hit up that sucrose, without going and checking any of the other water. Mm-hmm. Whereas the morphine, the THC, and the high mitragynine didn't necessarily like lock in on where the sucrose was, and they were exploring the other parts of the other water bottles a lot more. And you can see that sort of same banding differentiation happen when they moved the corner that had the sucrose in it. So like they were learning where it was, but then they switched the corner on them and they had to relearn where it was. Mm-hmm. Now, could they have just been like feeling loose and not sore, and their muscles were just sort of kicking, and they were just exploring more in general? So they're like behaviorally activated, and therefore they had more nose pokes in all of the corners of the IntelliCage. There seems to be like other interpretations you could make in that case, if we're just looking at like the percentage of correct responses in which corner and how long it took them. However, with the baseline treatments, activity and familiar cage, so like figure 2 sort of establishes that there wasn't necessarily that they were just exploring more corners in the cage. It was actually that they were just incorrectly guessing where the sucrose was. It's a cool study design, and I'm not knocking the study design at all. But so like when I say, could it be that they're just exploring more? Like the researchers obviously had thought about that and speak to that in the results. But in isolation, there's like other reasons you could postulate as to why they're seeing this behavior.
In that figure 4, it's like even the ones treated with morphine, THC, and high-dose mitragynine with that CB1 antagonists are all up there with the untreated mice in terms of place learning and reversal learning.
Well, I mean, that's the crutch of this entire paper, is essentially say, look, we see these impairments in the percent of correct responses to the correct corner, but if we give them the same treatment with the CB1 and antagonist, then we don't see that learning impairments. That's the main sort of take home, I think, of this paper. And what they were trying to establish is it CB1 receptor modulated, and at least maybe we don't know all the stepping stones between there, but it's certainly this behavioral effects were mitigated or extinguished with the antagonist.
And I guess they don't. They don't really test for other effects like pain, because I'm wondering what it would be like to, take morphine with a CB1 antagonist now. Like, would it be different, or even THC?
Right. So like if you did a tail flick test or a hot plate test, like how long it took them to move their tail off of something hot, that would be like a nociception.
And like, does it still feel the same, but you're more focused, your brain works better?
Or is CB1 even involved in the pain relieving effects, or is it just that CB1 receptor is involved in sort of learning and memory, especially in the hippocampus, and so it could have no effects at all? Like I hear what you're saying. Maybe with the CB1 antagonist, there would be either an enhanced like pain relieving effect or reduced pain relieving effect, but I don't think that there's precedent to necessarily say that the CB1 is involved in that angle of psychopharm.
So I think this is wrong, but does CBD act at all on the CB receptors? So I read something that said it's an antagonist, but I don't think that's right, is it?
Yeah.
Because I'm just wondering if it regulates the THC. Because I have my 1-1 tincture I take when I just want to relax.
Well, it's a little bit different than, so it's anti-inflammatory is like where the pain relief comes from. Whereas with opiates, it's like blocking the pain signals.
Okay,
and
yeah.
So I don't think it is an antagonist of CB1, but it certainly does have activity there, yeah.
Yeah, I was just wondering, like, if you could take CBD with some of these other things and wouldn't impair you cognitively.
I think in the context of being a CB1 receptor antagonist, it's like in the context of if THC is making you too high and like anxious and paranoid, you can take CBD and that will bind at the CB1 receptor but not lead to its activation, and it will like reduce the effects of THC activation at those receptors. Mm-hmm. But yeah, whether or not that would, so if CBD was an antagonist, much like this compound NIDA 4120, then would we see the same effects to the same degree? I don't know. It's a good, good hypothesis.
And I'm wondering, like, if you could take that NIDA 41020 and, like, if you get too high or something and you eat a high dose edible, and that would that make you not high? Like, I'm just wondering exactly what's going on with the mice. In terms of, like, cognitive impairment, does that just mean they're high? Or
I would assume that there's some degree of, like, similarities to what is CBD and this chemical. But, yeah, I would say, like, all of these chemicals that are just, like, given numbers are, like, research chemicals and certainly don't have very many, like, established use in humans and how that would potentially affect people.
Yeah. Well, it's pretty interesting because I didn't really know much about whether kratom or mitragynine acts on cannabinoid receptors. I've never really thought about it. I don't think we've talked about it before.
Well, I think it potentiates. I mean, we've known that if you take like opiate painkillers and you take it with medical cannabis, that like you're going to feel the effects of the opiates more strongly than you would if you were to just take them in isolation. So there's definitely like clear overlap and potentiation. And potentiation is similar to like the sensitization that they're discussing here, in that like it takes less amount of the active drug in order to feel the same effects. So we know that there's overlap there, but there's, of course, everything, the devil's in the details, and there's just, you get to the point where it's like, well, do we even know anything that's going on in there? I mean, we do, but it's never as clear cut as we'd hope it would be.
Mitragynine enhances resistant to punishment and sucrose seeking. This is air puff punishment, so I thought that was pretty funny. They put like a puff of air where there's
not much of a punishment,
is.
But I guess it'll startle you for sure. But like, it's not like an electric shock or something.
Yeah, that's what I thought it was going to be. And I looked at it, it was an air puff. Mm-hmm. So they say, screw the air puff, I'm high and I want some sugar.
And I guess when you ever like blow in your dog's face, they like, they tweak out when you do that. So maybe it's similar.
What is the immunostaining? I guess it's has something to do with figure 5 here.
So this is the antibodies that were washed over the brain tissue cultures, and the staining represents the CB1 receptors. So positive and negative controls, immunoreactive fibers in the molecular layer, a positive control. It's a positive control, negative controls. It's like they want to demonstrate that brain-wide, tissue-wide, where there are CB1 receptors, we're getting a staining, and where there are not, we're not getting as much staining. And then there's particular like brain area. They were looking at the hippocampus and the VTA.
Okay.
But these are like parts of the other brain, cerebellar molecular layer where there are CB receptors, but it wasn't necessarily what they were targeting in the experiment. I thought that, overall, in terms of like a lot of the papers that we have reviewed on the Journal Club, this is one of the best in terms of like the latest and greatest of behavioral psychopharm. they're using automated IntelliCage system. They're looking at the like metabolic profiles, or like protein expression and protein density, as well as genetic changes, up or down regulation, genetically. So it covers that multiple levels of analysis that I think is very characteristic of 21st century neuroscience research. So I definitely give them mad kudos on, number one, having the 100 mice, so having a thorough sample size group with very clearly delineated treatment groups, a high and low mitragynine, and then looking behaviorally and then down across those levels of analysis. So in terms of, if anybody wants to, any of the listeners want to dig into a paper, I would recommend this one. If you're thinking about getting into behavioral pharmacology, this is a really good article to sort of prepare you for the scope of what labs are putting out these days. And in several other papers, and it just has to be that way. But, I know that I've said of papers in the past, well, they should have added some behavioral, they should have added some genetic, just some of the sections in this paper could have been published on their own, but they waited, to collect all this data and submit it as one package, which I thought was very well done. I wish that there was a little bit less of the drug hysteria and just starting a sentence off blanketly by saying it's a widely abused plant-based drug preparation. It just seems to me politically motivated, not necessarily scientifically motivated. But we're not in the business of trying to pump those up or pump those down. I suppose we're just supposed to be reading the paper for what it is.
Yeah, and it's also worth mentioning they're in Malaysia, where kratom is still illegal, so they probably have different kinds of pressures there studying an illegal compound than we do here. They have a legal grow facility there. And I spoke to Darshan Singh. He's more of a social scientist. He was on the podcast before, but he is at University of Science Malaysia. But the thing is, he's done, because I'm trying to think of anywhere where there's evidence of cognitive impairment in social science studies, because I think he's done a study on long-term kratom consumers, and it found that they had no there wasn't any more psychosis in the sample size than there would be normally. So it was kratom doesn't appear to induce psychosis. And I think he's also done cognitive impairment studies of long-term kratom consumers and found that there was no significant cognitive impairment in the group that he looked at. So I'm kind of trying to link this to what it is in the real world, and I don't know, maybe it comes out in a different way that's less obvious.
I was going to say too, cognitive impairment in humans, might as well be a spectrum of 4 or 5 different cognitive attributes. So that's why I was sort of trying to be clear at the beginning about this is about the rate of learning, more specifically the number of correct responses in choosing a sucrose-based preference. It's more learning and memory and acquisition of behavior and rewarding or extinguishing of that behavior. It's a proxy for cognitive impairments, but of course, when you jump from mice to humans, what constitutes a cognitive impairment is different things and open to interpretation, certainly.
Thank you, Dr. Jonathan Cachet. Check him out on social media at jcachet. Check us out on Twitter at Kratom Science and on Facebook. Please subscribe, rate, review the podcast. The music is Captain Big Wheel. The song is called Moonrunner. Kratom Science Journal Club is produced by me, Brian Gallagher, for KratomScience.com. Take care.